Hey ANPers, this is Dr. Jones checking in. This is ANP1 Lecture Test 5 Video 4. And we're starting in on the middle of page 5 with section 12.2 to talk about neurons. So here is a picture of a neuron. It says there are 4 general regions of a neuron. We have the cell body, which is the soma. And that includes 2 parts, the nucleus and all the stuff around the nucleus, which is the peri, which means around, and caryon. So it sounds like a paracaryon luggage, paracaryon, but it's not. It's the stuff around the nucleus. So it's like the donut around the donut hole, except 3-dimensionally. So the donut around the jelly filling. Or like the solid crust of the earth around the molten liquid core or something, 3-dimensionally. So together they make the soma. The dendrites, also called the little bits that it's pointing to here, are also called dendritic spines, but we're going to use them as if they're the same thing. Dendrites, dendritic spines. D-R-I-T-I, dendritic spines. Anyway, they receive information. Receive is like I after E, except after C. I before E, except after C. Anyway, we're going to receive information. We're going to abbreviate because I can't remember how to spell the word receive. So this receives information from other neurons. Have we talked about the word synapse? Can y'all see the green on the tan? The synapse is where 2 neurons communicate. And so the dendrites receive information from another neuron. And then, of course, the axon, which sends information. I should have stopped at O, huh? Yeah, I totally could have just stopped at O and had info again. There was room. Okay, so the dendrites receive information. The soma does the information processing-ish. We'll get to that. And then the splits at the end of the axon. Now these are all terms, okay, these are terms we've had before. Paracaryon is new. This one is also new. The telodendria are the splits at the end of the axon. But they function like the axon. It's kind of like, this is my arm, but these splits at the end of my arm are called fingers. And they do have slightly different functions, but they're mostly part of my arm. Okay, so that's the same with the telodendria. They're mostly part of the axons, but they are splits. You recall we talked about the motor neurons and the motor units and how each motor neuron controlled a number of different skeletal muscle cells. So it'll be these telodendria that will split off to go to the separate skeletal muscle cell, even though each neuron only has 1 axon. So that's how it's able to have a motor unit. So where are the neurotransmitters made? Let's go to red. Okay, and the neurotransmitters are made in the paracaryon, which is this part around the nucleus. The Nizzle bodies are bits in the paracaryon, and they are clusters of RER and free ribosomes. And they stain dark, the stain that they usually use. They're different stains, but the stains they tend to use on brains, the Nizzle bodies stain gray. And that's why the soma of a neuron is generally considered gray matter, because it stains gray on the slides, specifically the Nizzle bodies in the somas. Okay. Now, since most neurons lack centrioles, there's no centrioles. Oh, centrioles. That was like test 1. Chapter 3. That was like months ago. What's a centriole do? Oh, wait, look it up. Pause, look it up. What's a centriole do? That's right, a centriole does cell division. Most neurons don't have centrioles, so can they divide? No, because without the mitotic spindle to pull the chromosomes apart, cell can't divide. So no cell division, which means that limits the response to injury and disease. So the lack of centrioles does have a consequence. All right, so and then we will draw, and here's a better picture. You've got the dendrites, dendritic spines or branches, and here you see the Nissl bodies, which are the RER and free ribosomes. You've got lots of mitochondria, of course. The axon hillock is the first part of the axon, and it's the part that specifically does the information processing, which we will discuss later once we have more terms and concepts. Okay, there's people right outside my house. Okay. And then the initial segment of the axon is this next part, just the first part of the axon. And of course you have Golgi, we kind of skip those, the nucleus and nucleolus, and the dendrites with the dendritic spines. And then for the other half of the picture, the axon, the cell membrane of the axon, just the axon part, is called the axolemma. And the stuff inside the axon is sometimes called axoplasm. So you've got your cytoplasm and your sarcolemma, plasmalemma type term. And then, of course, the telodendria we discussed, and they end at synaptic terminals, which are also called synaptic knobs or axon terminals. So now let's draw our own little version of that. That's not the one we want. Is that the one we want? White screen. All right. So I can't draw the tree branchy things, so I just simplify and do kind of a Lisa Simpson hair thing. And I'll just draw a rake with knobs at the end. Okay. So these are the dendrites, and what do dendrites do? They receive information. And I made it plural, so now I have to label another one. This is a nucleus. And then we'll draw a mitochondria, have an outer membrane and inner membrane with folds called cristae and stuff inside the inner membrane called matrix. So this is a mitochondrion. N. That's an O. Mitochondrion. And then we need our Nissl bodies. Nissl bodies are RER. RER are little loops with lots of dots because the rough endoplasmic reticulum has the fixed ribosomes. But we said that Nissl bodies were RER and free ribosomes. And Nissl was the guy's name, so we will capitalize it. Let's see, we can put a nucleolus in there because we've got room. Ooh, what's a nucleolus do? Do you remember? The nucleus contains the DNA, and the nucleolus makes rRNA and combines the rRNA with proteins to make ribosomes. That's right. Nucleolus. Nucleolus. Oh, and then color change. this part along is the soma, or cell body. They change the terms every now and then. I can't remember if the new one's soma or the new one's cell body, but they mean the same thing. And then these two together, no, are the soma. I did the wrong term. What was I supposed to put there? That's right. That's the parakaryon. K-A-R-Y-O-N. Parakaryon luggage. What I forget. Okay, this is an axon. This bit here is the axon hillock. In lab, it's orange. It does the information processing, and the axon sends information. This next bit of the axon is the initial segment of the axon. And then the outer edge of the axon is the axolemma. I forgot the O. The axolemma has voltage-gated ion channels that propagate the action potential. Yeah, write that down. The axolemma has voltage-gated ion channels that propagate the action potential. True story. For reals, though. And then the splits at the end, the little fingers, telodendria. And they also have voltage-gated ion channels, and they also propagate the action potential all the way to the, and we need all the terms here. What are all the terms for the little pads at the ends of the fingers? We've got axon terminals, synaptic terminals, and one more, going up because I'm out of space, synaptic knobs. And those all mean the same thing, okay? The little, like a gecko toe has a little swelling at the end of the fingers, like that. Geckos have the cutest little toes. They like hook onto the glass and you can see like the, so cute, so cute. Okay. Let's see, let's look at the note set and see what else we are supposed to talk about. Okay, we will talk about this top of page 6, which on my note set looks like this. Okay. The dendrites on a neuron receive information. You're right. Oh, I spelled it right there. Yay. Thanks to chemically gated ion channels in their membranes. I would highlight that part because that's a basic concept that you're going to need to know for, again, the entire rest of the semester. So the dendrites on a neuron receive information thanks to chemically gated ion channels in the membranes. The axon is propagating an action potential thanks to voltage-gated ion channels in the axolemma. Highlighter alert! Highlighter alert! It's time to highlight. Okay. And axonal transport is the new term. I've still got axoplasmic transport in my head. And that's moving stuff from one end of the axon to the other through the axoplasm. Recall the axoplasm is the cytoplasm in the axon. And so there are two directions to go here. We need a different color. Thank you. Ooh, magenta. Okay, so from the body down to the synaptic knobs is anterograde flow. So we already mentioned that the parakaryon makes the neurotransmitters. This takes forever to write out the words. It makes the neurotransmitters, and they are moved down the axoplasm via anterograde flow from the parakaryon, where they're made, to the synaptic knobs, where they're released into the synaptic cleft. So that is anterograde flow. Moving stuff from the soma to the synaptic knob. Going backwards the other direction, which I should totally do a different color. Do we have green on this picture? Great. Green. Going backwards from the synaptic knob up to the soma, or cell body, is retrograde flow. Retro means back or behind, like a 50s diner. Retrograde flow, retrograde axoplasmic transport in the previous versions of the book. Now it's flow instead of axoplasmic transport, but you know. And so that's moving stuff from the synaptic knobs backwards up to the soma / cell body. I forget which the new one term. I think cell body's the new term. And soma's the description. It used to be the backwards, but you should know both. So give some examples of stuff that can travel via anterograde axoplasmic transport. And that'll be stuff like neurotransmitters, enzymes, things that the cell makes that it wants to release. Stuff that travels via retrograde axoplasmic transport or retrograde flow are the things you don't want. So rabies, other rabies virus, heavy metals, bacteria, other viruses other than just the rabies virus, they enter at the end of the neuron and get moved up to the soma. So yeah, the retrograde flow is usually stuff we don't want. So. All right, so we will start in the next video with the next bit, the classification of neurons, which will be the next picture. Let's double check. Yeah, that's the next picture. Okay, so this has been ANP1, lecture test 5, video 4, where we covered the beginnings of section 12.2. Thank you, as always, for tuning in. The education must go on. Dr. J out.